us an opportunity to organize this webinar. I would also like to extend happy welcome to our Honorable Vice Chancellor, Professor K. Sivaramakrishna Sir, for accepting our invitation. I would like to extend welcome to our Pro Vice Chancellor, Professor Shivaprasad Sir, Registrar Sir, Professor K. Balaji, principals, directors of all the institutes and schools of Vizag, Hyderabad, and Bangalore campus. I extend welcome today speakers Dr. Thiagarajan M, faculty, senior lecturer from International Medical University, Kuala Lumpur, Malaysia. Dr. Gangaraju, assistant professor, faculty, School of Science, Hyderabad campus. Dr. Arvind Kumar Suri, CEO, tunsmiles.com, for accepting our invitation to this webinar. I am very happy to welcome all our participants from academic, industry, and community background for coming up from, for attending this particular webinar, for registering from different states. We are happy that 9 states have been registered from different states. So students, research scholars, faculties, all have industry personnels have joined this particular webinar. So we are very happy to extend a warm welcome to each one of you. In this pandemic situation of coronavirus, we know the importance of drug development, which is often very time consuming and a very expensive procedures. A proper idea about the drug delivery system will make the entire process of drug development process very fruitful and very successful. So today we all have gathered here on this online platform for this webinar session to attend on the topic of trends in drug delivery system by our reputed speakers for today. So we are happy to introduce this webinar session. So as we are all in the lockdown session, lockdown rooms, but our ideas are opened up, knowledge can be shared on this platform. So I welcome once again all of you for this complete webinar session, which is conducted by the Geetam Institute of Pharmacy and School of Pharmacy from Hyderabad campus. Now, I would like to extend the scientific session start from this particular time. Thank you.
Thank you, sir. Thank you for your kind words. So this is Dr. Sinoy. I would like to request Dr. Rakesh Barik to introduce today's first speaker. Sir, you can have the mic, sir.
Thank you, Dr. Shinoy. I hope I'm audible, clear. Hello.
Yes, you are audible.
Yes, sir. Good morning, esteemed guest speakers for today's session and honorable participants across the country. Our first speaker for today's occasion is Dr. Thiagarajan M, currently serving as Senior Lecturer, Department of Pharmaceutical Technology, School of Pharmacy, International Medical University, Kuala Lumpur, Malaysia. Sir is a PhD from the Department of Pharmaceutics, Yenungam University, South Korea, which he completed in the year 2014. His research work is mainly focused on development of liquid crystalline nanoparticles for enhancing topical delivery systems, especially the 5-alpha reductase inhibitors. He is involved in various research projects focused on lipid-based nanoparticle system for improving the delivery of poorly soluble drugs. His research areas mainly include to the liquid crystalline nanoparticles, solid lipid nanoparticles, liposomes, polymeric nanoparticles, etc. Various drug delivery techniques like the sustained, delayed, and the controlled release technologies, HPLC method development and validation are also coming in the domain of his expertise. He has published numerous research articles in reputed journals of international level and has presented his work in various international symposium and conferences. He is also a reviewer in various journals and is actively involved in multidisciplinary research and collaborative scientific works. So with this brief profile, I humbly request the first speaker of today's session, Dr. Thiagarajan, to kindly start his presentation.
Dr. Thiagarajan, sir, you can unmute yourself, sir. Yeah.
Sir. Unmuting and button, those things are open. Unmuting is open.
But I cannot hear your voice.
Yeah, we can hear your voice.
I think no one is hearing the voice.
No, you can, now we
Yes,
can
sir.
hear you.
Yeah, you're audible, sir. You're clearly audible.
Yeah, कोई मैम अच्छा ठीक है मैं बीच में ये कनेक्ट करो ना आई विल यूज आप एक काम कीजिए फिर से लॉग ऑफ करके लॉग इन कीजिए ओके आई विल ट्राई टू या एडमिट यू इफ यू आर कमिंग व्हाट्स योर नेम व्हाट्स योर नेम अच्छा ठीक है नो दैट इज नॉट अ इशू आप ये कीजिए
Dr. Sinai, I think
Yeah.
Dr. Tyaga is logging in. Just
Okay.
2 minutes of time.
Okay.
Yeah,
Okay.
is not able to connect. So just
Okay.
there is an session for 2 minutes.
Yes.
Yeah.
Yes, sir. So please, everyone, yeah, sir is going to log in a short while. Just the net issues, internet issues are there. So please be here.
Yeah, please. Yes, just now I introduced that. No problem. You start. No problem. Sorry.
So yeah, I think sir came actually. So yeah, Dr. Thiagarajan.
Yeah.
Yes, sir. Now
there is a technical glitches. Apologies for that.
Yes, sir.
Could you enable me to share the screen?
Yeah, you can share it, sir, because you are actually co-host now. Okay. So otherwise, yeah.
Right.
Now it's, yes, sir, please.
So you can hear clearly, right? Okay.
Yeah.
And see the screen as well.
Yes, sir.
Right.
Yes.
Sorry for the. First I would like to apologize for any technical glitches that happened just now, and it is amazing to be in this virtual platform with nearly 500 participants throughout our nation. And I would like to thank Geetham University for giving me this wonderful opportunity to talk on the current trends in topical and transdermal drug delivery. All right. Okay. So without further delay, I would like to start my presentation. And first, okay, why we are moving towards the topical or transdermal delivery is because the oral route or the parenteral route, when it fails to deliver certain essential drugs like proteins or some other small molecules, even which is having very potent effect, therapeutic effect. So we choose topical or transdermal drug delivery. And it is having huge potential in the market as well. Okay. So with that introduction, I would like to start my presentation. So this is a brief content of my presentation. As I was informed that most of the participants will be like undergraduate students. So my talk will be focusing on the basic elements of topical and transdermal delivery and what is the relation between the skin physiology and the permeation mechanism of the drug through the skin. And we will see the recent advances in the skin delivery like microneedles and other advanced drug delivery for the transdermal like using interphoresis or electroporation example. Okay. And finally, I will give a brief aspects on the hand sanitizer when we are using in our daily life at this unprecedented situation during the pandemic. So recently we have published a review article on this hand sanitizer, and I will be touching the basic components of the formulation of hand sanitizer. Yeah. Okay. Right. Yeah. So what is a topical delivery or a transdermal delivery? So usually we apply the topical formulation into the skin to treat the local disorder. Right. And the intention of the topical delivery is to retain the active pharmaceutical ingredient within the skin. So meaning that the topical, we want the formulation to deliver the drug to retain on the superficial layer of the skin, and we don't want the drug to reach the systemic circulation. For example, we have certain creams, for example, hydrocortisone cream or some other gels, ointments, all does the same function. And we can classify the topical into two broad categories. One is locally acting and regionally acting. When you say locally acting, we want the topical dosage form to treat the skin disease like which is superficial on the skin. It may be like acne or kind of psoriasis or any other lesions or any other skin disorders that occurs in the superficial layer of the skin. When you say regionally, for example, the person is having a knee pain, you are applying a topical cream or an ointment that contains pain relieving medication. So in that case, the drug has to cross the skin and then it has to penetrate to the deeper muscles to act. So that is regionally active. Okay. Whereas the transdermal is designed to deliver a given dose of drug through the skin to the systemic circulations. So when you say systemic circulation, the drug will permeate through the skin and then it get into the blood circulation and it goes to the systemic circulation where the drug reaches its site of action for the therapy. So the real challenge in both the cases, whether it is a transdermal or a topical, is the skin permeation. We have the superficial layer of the skin that is a natural barrier which will prevent the drug to penetrate to the skin, which is a natural barrier. Right. Okay. So here is a small image that differentiate the topical and transdermal patch. So for topical, we want the drug to be in the superficial, whereas for the transdermal, we want the drug to reach the systemic circulation. So the image shows the cross section of the human skin. So the human skin, the superficial layer we are saying as epidermis. The layer beneath the epidermis is the dermis. And once the drug crosses the epidermis, it can easily penetrate into the deeper membrane of the muscles, and then it reaches the dermis. So epidermis is the natural barrier we are having. That too, in the epidermis, the stratum corneum is the barrier that limits most of the drug permeations. So in the epidermis, we have stratum corneum, stratum granulosum, stratum spinosum, and then stratum basilae. Okay, so if you see in the microstructure of the stratum corneum, the stratum corneum has corneocytes and the lipid, the skin lipid. So it is like a mortar and brick model. So mortar is covered by the brick. So when you say mortar, it is the skin lipid, and when you say the corneocyte, it is the brick. So it is having a unique structure that prevents the drug to cross this membrane. If you further see into the aspect of what is actually present in the lipid and in the bilayer, it is the ceramide, and other constituents are the free fatty acids, cholesterols. These are the things. Okay. So now we need to have a clear understanding on this particular skin physiology, because we have to relate our dosage form design to overcome this skin barrier. Right. So as it contains most of the skin lipids, if for a drug which is having more lipophilic in nature, it can easily cross the skin barrier. Whereas the compound which is of more hydrophilic in nature, it finds very difficult to cross this membrane. And apart from the skin lipids and then fatty acid that present in the stratum corneum, it is also having certain amount of water. We say like transepidermal water. Right. For example, like moisturizing cream that maintains this transepidermal water to maintain the normal skin texture. Okay. Right. So we need to have a clear understanding of the physiology of the skin for a better or improved transdermal product development. It's not only apply for the transdermal product, it applies for all the other routes as well. So the first thing is like understanding the clear physiology of that particular site. Okay. So having said that, we will look into the different mechanism of drug permeation. So we can classify into three broad category of the drug permeation mechanism. So one is intracellular route, and second one is the intercellular, and third one is the shunt route. So in case of intracellular route, the drug from the formulation first is released, we can say like dissolution. Then once the drug is dissoluted from the formulation, it reaches the superficial layer of the skin, and it get partitioned into the corneocyte, and then it diffuses through the lipid and then reaches the viable epidermis. So once the drug reaches the viable epidermis and then dermis, it can easily absorb through capillary, and then it goes to the systemic circulation. So in case of intracellular route, it purely depends on the physicochemical property of the drug. If the drug is more lipophilic in nature, it can follow the intracellular route. So in corneocyte, it diffuse and then it get partitioned between the lipid. It's like a straight route, or the straight route of permeation through the skin for the intracellular route. Whereas the intercellular route, the drug will not diffuse through this corneocyte. Instead, it takes...
So Thiagarajan sir, sorry to interrupt, sir. Could you please disable the annotation? I think you have in that setting, in more setting, you can stop participant annotation, because people are drawing in between on the screen. Actually.
Yeah, actually, I also surprised to see all these things. Sorry for the interruption.
Yes. I'm sorry for that, sir. And I
So
request all the participants, please don't annotate on the screen. Okay, thank you, sir.
okay, okay. Now I stop that. Okay. So yeah, if you have any technical issues, you can rise at any time. Okay. Yeah. Thank you. Right. All right. So I stopped at intercellular route, right? So I continue from there. So intercellular route, it follows a Tartarus route where the drug is partitioned into the lipids between the coniocyte, between the coniocyte, and it diffuses through the lipid, which follows a Tartarus route. And then once it reaches the viable epidermis, it can easily absorb through the blood circulation. And the drug which is having like a considerable lipophilic character, it can cross through, it can follow this intracellular route. And the next route is the shunt route. In the shunt route, it follows the sebaceous gland route, or most prominently, it follows hair follicle route. And nearly like 1% of the drug that going to the transdermal route is following the shunt route. So in case of shunt route, the drug just goes into the hollows of the sebaceous gland and the hair follicle. Okay, right. So far we have seen the different mechanism of drug permeation. Right. So next, we will see what are the ideal characters for a drug to have can be formulated as a transdermal drug delivery. So the first one is the molecular weight. If the molecular weight is lower than 500 Dalton, it is a good candidate because it can easily cross the skin membrane. And secondly, if the drug dose is low, but still it is having higher molecular weight, still it can be can be delivered through the transdermal route, right? And second thing is the partition coefficient, optimal water partition coefficient. So which is having in the range of, the drug is having the range of 1 to 4, is an ideal candidate for the drug delivery. So despite of this, even if the drug is more lipophilic in nature, it can be also effectively delivered through the transdermal route. And the other parameter or the factors considered in drug formulation is the dose. So if let's say like we compare the topical or transdermal route with the oral route. So oral route, we can take like 500 milligram of dose. So whereas in case of, imagine about the patch, it's very difficult to formulate such a high dose for the transdermal delivery, right? So the dose should be as low as possible for a transdermal delivery. So, and which will be having a good.
Sir, sorry to interrupt again, sir. So this is the thing is that can you reshare? Can you start the resharing again? Sharing, because I think the old that graph means whatever the annotation people were doing, it is still there. So for some people, it's not clear actually. Okay.
Okay.
I'm sorry to interrupt,
So
but
that's okay.
Okay, sir.
Okay, right.
Yeah, now clear, sir. Yeah.
So now it's clear. Okay.
Yeah, it's clear now.
Okay, thank you. Right. So, right. So the dose is another parameter we need to optimize. So typically, it should contain like 1 milligram per centimeter per day is an ideal candidate for a transdermal activity. Right. And for any formulation, topical or a transdermal formulation, the traditional one is of using a certain permeation enhancer. So the permeation enhancer has the property to alter the organization of the lipid bilayer. Just now we have seen in the superficial layer of the skin, which is the stratum corneum, right? So this penetration enhancer can causes some changes in the lipid bilayer, and thereby it improves the drug delivery to the skin. Right. So either it interacts with the intracellular protein or like altering the partition of the membrane, or combination of these mechanisms. For example, the figure shows different mechanism of permeation enhancer. If it is a polar or lipid enhancer, so it mixes up with the lipid bilayer and then it causes a membrane disorganization. So if it is a polar enhancer, it increases the fluidization by combining with the polar head group. And similarly, the other lipid extractions or the phase separations or inner. So these are the different mechanism of permeation enhancer. So depends on the type of permeation enhancer we are using, we can achieve an increase in the fluidization of the stratum corneum that result in the increase of the drug permeation through the skin. So mostly we use alcohols or fatty acids, and other classical example are ozone and some other lipids. Right. So all these will mix up with the stratum corneum and alter the skin permeation. Okay. Right. We have seen the skin permeation enhancer. So now we will see what is there in the transdermal patch. So in any transdermal patch, the first thing is like we are controlling the dose of the drug. We are controlling the release of the drug so that we are achieving a constant delivery of the drug to the skin that diffuse through the skin membrane for the systemic therapy. So due to the skin barrier properties or like skin permeation limitation, so only we have handful of molecules that can be successfully formulated into a transdermal delivery. So for designing a suitable transdermal patch, we need a clear understanding on the drug physicochemical property, the drug release through the membrane, and then we need to quantify how much amount of the drug that has been crossed the membrane over the period of time we save flux. It's called transdermal flux. Right? Okay. So these image shows different types of transdermal patches. The first one is a simple patch where we are having just a drug reservoir. So in this case, mostly we use most hydrophobic nature of the drug, so that can easily cross the membrane. So the second one is the drug in matrix. So it is having a good control of drug release because we are formulating the drug with certain release modifying polymer, which is made into a matrix. And third one is the rate limiting membrane type, so which combine the drug in matrix as well as a rate limiting membrane. So where we can achieve a better control on the drug release. So all these patches has their own advantages and disadvantages, but ideally what expected is we need to achieve a required therapeutic concentration of the drug in the blood plasma over the period of time. For example, we have nicotine patch, we have fentanyl patch, and other contraceptive patches. So these patches has the drug reservoir as well as rate limiting membrane type patches that delivers the drug over a period of like maybe 1 week or even higher than that. Okay. Right. So far we have seen the traditional way of developing a transdermal or topical dosage form using skin permeation enhancer or a patch that controls the drug release. Okay. So now I will give a brief overview on the recent technologies to kindle some of the interest, those who are embarking their research in the field of topical or transdermal delivery. And broadly, we are classifying the transdermal technologies into active and passive methods. When you say active methods, we will be using certain physical, external physical equipment along with the formulation to achieve a better transdermal flux. Transdermal flux in the sense like improved skin permeation of the drug. Right. For example, when you say microneedle, the microneedle will, once applied on the skin, causes a physical, minimally invasive damage to the skin, and thereby it breaks the stratum corneum barrier and then enhance the skin permeation. So in case of microneedle, the device itself is creating a hollow space on the skin. Then we are applying the formulation. The formulation will have a certain desirable drug releasing property to improve the skin. So here it's a combination of the formulation as well as the device. Whereas the passive methods, it's solely focusing on, or it's only focusing on the formulation, like for example, liposomes or using chemical permeation enhancer. So the eutectic systems or products, these are the conventional way of improving the drug deliveries, which we say as a passive methods. Right? Okay, so the other active methods apart from microneedles are using thermal methods, which uses laser techniques. So in case of laser techniques, we can precisely control the intensity of the laser that can remove the stratum corneal barrier precisely on the skin. It's like similar to the laser technology which was used for the beautification to remove the wrinkles. Right. So what happens is when the laser is passed on the skin, it removes the stratum corneum. Then once we apply the formulation, we can achieve a better permeation effect. And the other method is using the radio frequency. So in both laser or radio frequency, it alters the skin permeation property or the skin property of the stratum corneum. The second one is the electrical method. In case of electrical method, we are using electrical potential or using the electric field, we are causing certain poration on the skin to improve the skin permeation. We have seen the microneedle, and the other few techniques are using the jet injector or ultrasound. So first we will see about the iontophoresis. So what happens in the iontophoresis? So iontophoresis, we have drug reservoir, which is the anode, and the other electrode is the cathode. Right. So this particular iontophoresis is a very helpful technique for delivering small molecules of particularly the charged species, which is more hydrophilic in nature. So you know the hydrophilic drug is always having difficulty in crossing the skin membrane. Right? So when you are loading the charged species of the drug on one electrode, and when we pass the electric current, this electric current is acceptable by the skin, meaning that the voltage or the amount of current will be very low, so that it can be accepted with the skin. Right. So once you apply the voltage, right, there will be a movement of the drug molecule through the skin. The amount of voltage or the extent of voltage can be used to regulate the flux of the drug molecule through the skin. And the time duration is another factor that decides the amount of drug that reaches the skin. So commercially, we have phoretic or lidocyde. So you can see on the image, like there is an electrode where you can apply the dosage form, and then using this particular controller, we can precisely regulate the amount of current that can be passed to the electrodes. And there is another example which is lidocyde. So both are very useful in delivering the drug based on the iontophoretic principle. Right? And the other methods are like the liquid jet injections or epidermal powder immunization. So in both these cases, what happens is like we are supplying a very high velocity of a thin stream of the dosage forms, so particularly useful for immunization purpose. So the dose of the active molecule will be very minimum, like even microgram range. So the basic principle is it's similar to the parental dosage form. So in parental, you are using certain diameter of the needle, right, to poke the skin. So in this case, the diameter of the stream is very lower than minimum needle, and because of the high velocity, it is having the property to cross the skin permeation, skin membrane, and then it reaches the deeper layer of the skin. And in case of electroporation, The electroporation involves, again, similar to the iontophoresis, but in this case, the electrical potential is used to create certain pores, or we say like ablation, or it is like mashing the tissue. Right? So due to the electrical signal, it causes some pores which can use to disorganize the stratum corneal barrier or increase the fluidization of the stratum corneal barrier. Thereby, once you apply the once you apply the formulation, it improves the skin permeation. And the next one is the microneedle. And in the recent past, there are a lot of research has been conducted on the microneedles, and it is easy to fabricate, as well as there are a lot of patents coming on this particular technology. And the advantage with the microneedle is it is minimally invasive, and it can, and particularly useful for the delivery of certain proteins and even the immunization purpose. So microneedle we can classify into 4 types. One is solid microneedle. So in case of solid microneedle, it is like just poke and then apply the formulation. The drug will reaches the systemic circulation. And in case of coated microneedle, we are coating the formulation on the surface of the needle, and then we are administering. So in case of dissolving microneedle, we are using certain biodegradable polymer, and the biodegradable polymer is loaded with the drug. And then once you apply, the needle stays in the skin, and then slowly the biodegradable polymer will degrade and release the drug. And the last one is the combination of the solid and the dissolving microneedle. So the last one will deliver precisely to the deeper layer of the skin. Okay. Right. So we have a commercial formulation on jet injector, which is the T-jet. So which is used to deliver somatotropin hormone. Also, we have the other microneedle-based products like Intenza, which is used for the immunization against influenza, and the other microneedle. Okay, so all these devices or the technology cannot function alone. So we need to design the formulation and combine the formulation along with this device. Okay. Right. So what are the challenges and what is the future perspective available in this particular area of drug delivery? Right. So we know that creams and ointments or the other dosage forms like gels are less effective for systemic therapy because of the poor skin bioavailability. Right. So when you say poor skin bioavailability, it is because like when you are applying a cream, you don't know how much amount of cream you are applying on the skin, whether it is cream or ointment, it is the same. So the dose is not accurately maintained, and it cannot achieve a better therapeutic concentration. Right. So in addition to that, there is a variation in the formulation application area. So what is this variation in the formulation application area? So our skin is differ in different body size. For example, if you are applying on the arm, the skin nature is different, and you are applying on thighs or the other parts will have different skin permeation ability. So in that case, if you are designing one particular transdermal dosage form, when you are applying on the different site, there may be a chance that the bioavailability cannot be achieved. And moreover, with the ages, the skin permeation, the skin property varies. For example, the younger ones will have better permeations. The children will have more better permeations compared with the elder one because of the difference in the skin nature. Right. So all these factors are challenges in designing the transdermal delivery. So, however, like with a clear formulation development and design, can deliver the drug with a better partitioning into the stratum corneum with enhanced transdermal flows. So what is this formulation development and design? So just now I have shared with different delivery technologies, right? So we need to combine those design of the device along with the formulation development to achieve a better partitioning. So it requires a extensive research on how much amount of the drug has been crossed the skin membrane using various in vitro techniques as well as the in vivo techniques. When you say in vitro techniques, mostly we use a France diffusion cell to study flux of the drug molecule. Right. So in that case, we may use a rat skin or a mice skin, and porcine skin. Right. So each skin data will have a different value of the flux. And when you study with a human cadaver skin, the data may differ. Right. So these are the challenges. I mean, these are the challenges we have. Right. And having said that, so how to overcome these challenges? So the technology which can overcome the barrier, for example, like when you're using a microneedle, which can cross the stratum corneum, and when you're applying the formulation on the skin surface, so you will get an understanding of the flux, the drug, after the microneedle application and before the microneedle application. So in that case, you will have a clear understanding how much drug has been crossed through the skin. Right. So still this area is in the growing phase, and hope like there will be a lot of innovation coming on this. And further, the formulation development is again a key success. I mean, formulation development is the essential thing to achieve a successful transdermal delivery technologies. Okay. So next part of my topic presentation is on the hand sanitizer. Right. So in the recent past, I mean, regularly we are using all these type of hand sanitizer dosage forms. So I would like to just share a basic information on these. Right. Whether you can use a gel or a foam or cream or a spray. Right. So how effective it is. Right. It depends on which type of formulation you are using, and secondly, how you are properly using that. Right. So when you see as a pharmacist perspective, we can broadly classify those hand sanitizer into two types. One is alcohol-based and non-alcohol-based. Alcohol-based, we say ABHS, which is alcohol-based hand sanitizer. So the United States, the CDC, or most of the regulations recommend the usage of alcohol-based sanitizer in the range of 60 to 95% volume by volume of alcohol. And in case of alcohol-free, we have a set of active ingredients like chlorhexidine, chloroxylenol, or most widely used iodine or quaternary ammonium compound, and triglyceride. So apart from this, we have the soaps, right? And they recommend like first the usage of soaps. If soap is not available, then we can go for hand sanitizer formulations. Right. So all these alcohol-based or alcohol-free has shown certain effective against the coronavirus or other pathogenic microorganisms. Right? Okay. And this table shows the three formulation which is recommended by World Health Organizations as well as the CDC, which is or the USP. So in all these formulations, we are using ethanol or isopropyl alcohol in different concentration, along with hydrogen peroxide. And glycerol is used to, glycerol, the purpose is to prevent the dryness of the formulation, which is a humectant. Right. Okay. So with that, I conclude my presentation. And thank you very much for listening. So before I end my presentation, I would like to state a famous quote from our former president, Abdul Kalam. Right. So this message is to all the students. Like, so don't worry about the fail, because fail is the first attempt in learning, and there is no end because its effort never dies. And then if you get answer no, meaning think that it is next opportunity, right? So go beyond, try to achieve your dreams. And that's the end of my presentation. I would like to thank Dr. Shivkumar Sir and his team for this wonderful opportunity to share my research on the presentation. Thank you very much. So with this, I pass the session to the.
Yes, sir. Thank you, sir. Thank you, sir. Thank you, sir. Thank you for the informative session and for a nice talk. So I will now request Dr. L. Srinivas, sir, to please moderate the question and session from the participants.
Yeah.
So I think sir was here actually. So Professor L. Srinivas, sir, can you hear me?
Okay, I think there's some issue, some technical issues over there.
No, I think,
So,
sir.
yeah,
okay, I'm making co-host because his one ID is not clear actually.
Okay.
So, Professor L. Srinivas, sir, I have made you host now. Please, if you could hear, because your doctor version is not getting on actually. Okay. So if participants, if you have any questions, you can raise your hand. So I will unmute you, please.
Yeah. Few questions had come in my chat box.
Okay.
Like one of them, sir.
సమాజ అభివృద్ధికి తోడ్పడాలి ఏమో సార్ ఈ కుళ్ళులోనే ఉంటూ నేను సమాజంలో జరిగే అభివృద్ధిని నేను వెళ్తాను అలా కాదు రాము మొదట్లో నేను
Yeah, sorry, sir. In between, so if someone, if they can type their messages in the chat, we can ask.
Yes. Yeah, one of two of them have sent the questions. So, sir, one person wants to ask, what are the scope of using natural products, the herbal products, for these delivery systems, and what are the challenges that we face in that?
Yes, I got the question right.
Yeah.
So the use of natural compounds for the transdermal technology, isn't it? Right?
Yes, sir.
If I understood the question correctly, so how the transdermal technology can be improved using certain natural compounds?
Yes, sir.
Right. So whatever the principle, in the first place, like whatever principle that applies to the small molecules or the larger molecules still applies for the natural compound as well. So it depends on the properties of that particular compound. When you say property of the compound, whether it is a pure natural compound or it is like a crude natural compound, we say like crude extract, right? So ideally, both will have the challenges in causing the skin formulation, right? And for example, I take classical example, say curcumin, right? So curcumin you can see in several topical products. And even in most of our customs, we use curcumin just applying on the skin or whatever it may be, right? So if you relate the curcumin, right? So curcumin is a poor soluble compound as well as it is having considerable lipophilic in nature, right? So it can cross the membrane easily, and then that's why it's showing a yellow pigmentation on the skin, right? So the reason why I took curcumin is like it's a simple example to relate the property. And similarly, if the natural compound, we cannot, it's like a too broad or too general, the question is, right? If you say like one particular compound which can improve or alter the skin nature, particularly the natural compound, right? So we have to identify those novel compounds from the natural sources. Then we need, the next challenge would be like formulating that. Okay, so in a summary, whether it is a natural compound or a synthetic compound, the delivery utilizes or the development of the transdermal technology follow the same principle we need to have understanding on the property of the compound, and we need to have extensive study whether it is really improving the skin permeation. Okay. Thank you for the good question.
Okay, thank you, sir. Another question is from N. Venkatanagar. Okay, is it possible to deliver BCS class 4 drugs by the transdermal route?
That is another good question. When you say BCS class drug, it is having a poor solubility as well as the poor permeability. So the BCS classification of the system is mostly for like oral, we say, right? Like permeating to the intestinal membrane, right? So since this membrane, the skin membrane, is more similar to that, we will be having two challenges, right? The amount of the drug you're loading into the carrier, which will be the first challenge because it is having poor solubility, right? So poor solubility doesn't mean that the drug should get solubilized in an aqueous environment or a hydrophobic environment. So it should get solubilized in both the aqueous or and hydrophobic environment in a balance so that we can achieve a better therapeutic effectiveness, right? So that is the first challenge,
Okay, sir.
loading into a suitable carrier, right? So the recent technology, I think the next presenter will be presenting on the nano formulation, right? So there are a lot of technologies which can encapsulate or which can increase the encapsulation of the drug, which is having poor solubility. For example, like a solid lipid nanoparticle or nanostructure lipid carrier. I'm just giving an example. It doesn't mean that only this carrier can improve the solubility. There are many other carriers. So we need to have understanding on the property of the drug and the carrier which we are going to choose. So that will determine how much drug can be encapsulated, right? So that's the first challenge. And second, as you address the permeation, again, this carrier can improve the permeation. So for example, nanoparticle. The nanoparticle has the property to achieve a better encapsulation, and secondly, it can penetrate the membrane, and thirdly, it can also alter the drug release. So in that way, we can use modern technologies to deliver the class 4 of the BCS, drug which is of the class 4. But again, we are having challenges in selecting the right technology to achieve the better therapeutic outcomes. Yeah.
Okay.
Hello, sir.
Thank you, sir. Yes.
Hello.
Yes.
Yes,
Yes.
sir. Yes, sir.
Srinivas,
Srinivas,
we can hear you.
Sir, yes, yes. Thiagarajan, sir.
Yeah. Thank you.
Oh, hello. One small question, sir.
Yeah.
Regarding dosage considerations, what are the practical challenges we'll face in this system?
Sir, can you elaborate the question? Dosage?
Usually, dosage consideration in the transdermal systems.
Okay. Yeah.
Generally, we cannot administer complete dose in this. This is a practical
Yes.
consideration.
Yes.
So, what kind of considerations you have to take for fixing the doses? Because these are now becoming unit dosage systems, where you cannot exactly fix the unit dose like other tablets and all.
Yeah.
So what is
So
the practical issues?
Okay.
This?
Yeah. So that is a good question. Like even the challenges I have addressed are the consideration in the dose, like 10 milligram is an appropriate dose daily to achieve a better therapeutic outcome. So frankly speaking, that is one of the limitations with the transdermal or the topical dosage form, right? So for some of the drug, it is obvious that the dose is always higher. And when you want to design a patch, for example, even if it is like 100 milligram, right? Then
Yes.
you cannot, it is a challenge to deliver it. So however, some of the technologies where you can reduce the amount of dose and you can increase the transdermal flux. Okay, so what I am trying to say is you are delivering 100 milligram of the drug, out of which 50 milligram reaches the blood circulation, which is sufficient for the therapeutic action, right? With the technologies, if you are delivering the whole 50 milligram and the whole 50 milligram is reaching the systemic circulation, then you can increase the amount of dose. Okay, so that is a challenge. And second, as you address the permeation, then the technology has reduced the required therapeutic concentration of the drug that goes to the systemic circulation. So in that case, the formulation will have a 50 milligram of the therapeutic dose, and then since you have reduced like 50% of the drug to design a formulation, and you are achieving a 100% bioavailability. So all the 50 milligram is reaching the systemic circulation. So here the concept is the technology has reduced the loading dose, which is also achieving the required therapeutic concentration, right? But these technology are still in the it's not like completely explored, or you cannot see more number of products on the transdermal of this thing. And this is one of the limitation which you have addressed clearly, like the dosage consideration or the amount of dose that has to be delivered to the skin.
Yes, sir. Yes, sir. One more small question from other participants. Does the alcohol-free sanitizer will have same effect on applications as alcoholic sanitizer? Which one is more effective?
Okay, so that is also a good question. If you see the sanitizer, particularly the alcohol-based sanitizer, we are using 60 to 90% of the alcohol. And one of the major limitation with that is it's highly flammable. You apply the hand sanitizer and you go near any hard surface, there is a chance of flammability, right? Also, it alters the skin nature because alcohol is a good, right? So these are the drawbacks with the alcohol based. And the question was like the alcohol-free hand sanitizer, how it is effective, right? So there are very limited study on those aspects. For example, like chlorhexidine. Chlorhexidine is an alternate. It's an effective compound that can be used as an alternate for alcohol-free sanitizer. Right? So there are few studies that has been conducted or performed with the chlorhexidine against certain coronavirus, not the COVID-19, the previous coronaviruses like SARS-CoV or MERS-CoV, and the results have shown some promising results, right? But if you see the regulation in the point of regulation, still they are considering alcohol-based sanitizer, even in the hospital usage in the hospital settings or any other settings, right? And there is a good opportunity for us as a pharmacist. We can also look into that aspect. That's a good question. We can look into that aspect. Alcohol-free sanitizer is a promising, trend.
Okay, sir. Thank you. I hope there are no further questions.
Yes, sir.
I'm not seeing any other questions. Any other questions from the participants? I hope no. So now it's the time to conclude this session. I take the opportunity to welcome Professor S. Ganapati Garu, Principal, Geetham Institute of Pharmacy, Vizag, to give concluding remarks.
Yeah, thank you, Senior sir.
Sir, Professor Ganapati, sir. Sir.
So, Professor Ganapati sir, you can make yourself unmute actually. You are actually co-host.
Is it audible now?
Yes,
Yes,
sir.
sir. Yes, sir.
Yes, sir.
Yeah.
Good afternoon, sir.
Good afternoon. Thank you very much, Dr. Thiagarajan. For the last
most welcome, sir.
we have been locked and closed our minds, and your lecture again opened us, and we really enjoyed your lecture. And the lecture is highly meticulous, informative, lucid.
Yeah,
And
thank you so much for that.
these transdermal drug delivery systems is one of the best delivery systems, I always feel, because for children and old people, no, and you can easily deliver the drugs without any noise, no.
Yeah.
So I think our participants, they have benefited thoroughly with your lecture, and I thank you once again for taking your time, for sparing some time for us, for Geetham. And
thank you so much, sir. You're most welcome as well. And I was also really enjoying the session because this is like although it is virtual, I can see like nearly 500 participants are there, and I was quite amazed and then enjoying with it. Despite there is a technical glitches always we experience through the online platform. And initial part of the presentation was kind of technical glitches, and sorry for that. And then
it's a transformation now. It's a transformation now after Corona. Before Corona and after Corona, not before Christ and after Christ. All right. So there is a big transformation now, and we are all tuned to classwork teaching. And now online teaching means really it is new to all of us. And I thank you again. And please be in touch with us, with our colleagues. Some of our colleagues are doing good work on transdermal drug delivery systems. And
sure, sir, we will be in touch. I am also very happy to collaborate or extend my collaboration with the faculty.
Yes, sir.
Ethan. Yes, sir.
And I also thank our Shivkumar and his team for making it possible today to webinar lectures. So thank you once again. Thank you.
So just last note, Kumar sir, he's known for nearly 4 years. He was my previous colleague.
Your colleague in Malaysia, I think so. Yeah. Yes. Very nice.
And I think we stayed in the same apartment where currently I'm delivering the lecture.
I see. Okay. Yes. Thank you, sir.
Most welcome, sir.
Thank you.
Okay, sir. Thank you, sir. Yes, sir. Thank you, sir. So I request Rakesh, sir. So can we conclude now?
Yes, I think we can conclude.
Yes, I'm asking, asking everyone's permission to conclude this session. Thank you all. Thank you all.
Yes.
And
thank you, Rakesh sir, and Shinan.
Thank you very much, sir, for that session. Thank you so much.
And I request all the participants that whoever were not able to come in and whoever had some trouble, the recordings will be given to your groups, and we will also upload into the YouTube channel in our YouTube channel. Links will be provided later. And because today we could not stream it live. There was some technical issue from the Zoom side, so that's why. So so this is just one information, and the feedback form will be provided at during 5 to 6 p.m. Which is the last session. Okay, so that's also.
Rejoining at what time?
Rejoin at 2 o'clock. Please.
So you can use the same link, participants, same link which you are given in the morning for joining it at 2 o'clock for the second session. Okay, sir. Thank you.
Okay. Thank you.
Thank you.
One session ಆಯ್ತು ಅಷ್ಟೇ. ಈ ಟೈಮ್ ಇದೆ, 1:30ವರೆಗೂ ಟೈಮ್ ಇದೆ, 1:30-1:45 ಟೈಮ್ ಇದೆ. ಬರ್ತೀನಿ ಒಂದು ನಿಮಿಷ ಇದೆ, 5 ನಿಮಿಷ ನಂಗೆ ಟೈಮ್ ಕೊಡೋದು ಮುಗಿಲಿ. ನಾನೇ ಬರ್ತೀನಿ. ಪ್ಲೀಸ್ ನನಗೆ 10 ನಿಮಿಷ ಟೈಮ್ ಕೊಡು, ಬರ್ತೀನಿ. ನೆಕ್ಸ್ಟ್ ಸೆಷನ್ಗೆ ಸಾರ್ಟ್ ಔಟ್ ಮಾಡ್ಕೋಬೇಕು ಅದಕ್ಕೆ. ಹಲೋ.
Hello, yes, sir.
ಪರವಾಗಿಲ್ಲ ಸರ್, ಓಕೆ. Yes, sir. Correct, sir. Correct. Yes, sir. Yes, sir. So, yes, sir. Sir. అది 2 గంటలే సార్, 2 గంటలకి. సో ఇలాగే మనం కంటిన్యూ చేసేసుకుంటే 3 సెషన్స్ కంప్లీట్ అయిపోతుంది సార్, ఏమి ఇష్యూస్ ఉన్నాయి సార్. మధ్యాహ్నం అయితే ఇక్కడే ఉంటాడు కాబట్టి మనకి ఈవినింగ్ గానే ఉంటుంది కాబట్టి మన సౌండ్ కూడా ప్రాబ్లం ఉంది సార్. సార్ మధ్యలో జాయిన్ అయిపోతే అలా చేసేస్తాం సార్. ఎస్ రైట్ రైట్ రైట్. ఓకే సార్, ఓకే సార్. అదే ఇనిషియల్ కొద్దిగా ఇష్యూ సార్, బట్